Bismillahirrahmanirrahim. The topic that we are going to cover today is the responsible use of lab animals. And we are taking the course of good laboratory practices. And in good laboratory practices, the responsible use of lab animal is very, very important. So we have to use very responsibly all the animals that we are going to use in research. Mostly we are doing the research on mouse. So either we are going to use either like mice or rat, or we are using rabbits, or we are using the higher like animals. We are doing research on monkeys or chimpanzees. We have to do the research very responsibly, and that's also part of good lab practices that we should. And this is what we are going to learn today in today's lecture. That how we should use these animals, how the regulations are done, especially if you have to do the publications, what are the requirements that you have to follow if you have to publish that book. So the responsible use of lab animals, animal research plays a vital role in advancing scientific understanding and developing life-saving medical treatment. Like for my whole life, for the last more than 20 years, I'm using the lab animals for my research, either on therapeutic proteins, or I'm using the lab animals to do the research on stem cell. So for all these, we have to use the animals for our research, and they are very important. Like if we have to do the research on stem cells, and if we are like getting the stem cell from the human, but before that, like what could be the application of those stem cells? Either we are going just we are going to do some research, or we are going to use for therapy, or whatever is the purpose. We first have to do the work to optimize the protocol. We have to work on the animals. Like if you get the samples from, for example, from the bone marrow, it's a very lengthy procedure to do the bone marrow aspiration from the human. And if you like take 100 ml of sample to isolate the stem cells, and you don't have a good protocol for further processing, then you are actually wasting that sample, and there's also harm to the human. But in order to optimize the protocol, in order to test those cells, we do the research on animal. And this is like, as I mentioned, I am doing for the last more than 20 years. We are using those animals, optimizing the protocols, and then we are using those protocols on the human. So it must be conducted with the utmost care and consideration for welfare of the animal involved. Now, even like in the present era, there is a lot of movement now all over the world that we should use the lab animal very, very carefully. It's not like we have a protocol and we are like just randomly testing those on the animals. It should be very careful that we should choose the study and choose the animals, and we should properly follow the protocols for that what we are going to do, and then we proceed for further experimentation. And this is what we are going to learn today in our lecture. So why we use the lab animals, and why animal testing is important for our research. One of the things, this is what I'm also discussing, is advancing medical knowledge. So like all the protocols, all the procedures, because animals, they also have like similar enzymes, they have similar proteins, they have similar genetic makeup. So we don't use any drugs directly on the humans. First, we use those drugs on animals. And in order to like whatever study you are going to do, either like you are going to study, for example, you are going to study that how hepatitis happens, we can do on the animals. How we have to study some like neurodegenerative disease. This is like not directly on the humans, but we can do on the animals. One of the things is like neurodegenerative disease. They take years and years, like it takes 2 to 3 decades to develop those diseases. But in animals, for example, the life of the mouse is very short. It's like just 2 years. So the symptoms appear very fast, and then we can study those diseases uh, in like very fast. So this is the reason that we are using the lab animals, so we can advance our knowledge, advance our understanding about, like, if we have to do the diagnosis, we can use the animals. We can plan some study on the animal. If we have to do like treatment, then we also use those animals and study those. Even in the present era, we are using like research for regenerative medicine. So for regenerative medicine, in order to advancing our knowledge, we are using the animals, just like I mentioned in the very start. Then the number two is we have to ensure the safety. Animal testing help identify potential side effects and risk that must be addressed before a drug or product is used in the human. So, for all the drugs, sometime, for example, the journey start with the testing of the drug on the cells. So, what we initially do, we grow the cells in the laboratory and then we test the drug. And like if we are, for example, if we are targeting certain uh, enzymes in the cell, and the drug has proved to be very successful in the cells study, then we proceed with the animal trials. So sometimes drugs behave very differently in the in vitro study than in vivo study. So when we go on the animal trials, the result may be totally different. So results like the drug that proved to be like not very toxic for the cells may prove toxic for the animal, and that's why we are not like directly using all the drugs on the human. So it's like the pathways we test on the cells, then we go to the animals, and then we go to the human. So to ensure that our drugs are very safe for the humans, so we, in order to ensure safety, we go for the animal trials. And then there is ethical consideration. So rigorous review process and strict guidelines help ensure animal research is conducted ethically and with minimal suffering. So now there is like a lot of uh, uh, focus is on ethical consideration. So whatever study you have planned on animals, it go through the ethical review board and they approve those studies and then we perform those studies. It's not like if some study is in your mind and you're working in some institute, you can't start directly that study. So it should go to the ethical review board, and then we proceed with the study the uh, in the animals. So, what are the ethical consideration in animal research? So, one of the very important thing is we have to minimize the harm. So, it's as the name indicate, we have to minimize that harm. There should be less and less harm to the animal. So, research must be carefully considered the potential benefit of the work and strive to minimize any pain or distress caused to the animal. So, this is like very very important now. Whatever study you are planning, we should try that there should be minimal harm to the animal. And then there should be focus on replacement and reduction. So we can replace the maximum we can replace the animal. This is what we are going to learn in our future slides, that how we can replace the animals and how we can reduce the number of animals. So, like by carefully planning, we can definitely replace the studies. And in a whole slide, we'll see how we can replace the animals and how we can reduce the number of animals. And the third and important thing is we should respect all the life. So animal also have the life, so we have to respect those. And that's why we should go for like replacement and reduction in the number. And we should try our best that there should be minimum harm to the animal. So this is like ethical consideration. Then regulations and oversight for animal welfare. Uh, First, if we look about the regulatory oversight, there should be strict law and guidelines to warn the use of animals in research, ensuring their human treatment and well-being. So, uh, like this is now being ensured in almost all the developed countries. So in all the developed countries, whatever research you have to do on the animals, there are very strict guidelines of working with animals, like all the care, where you have put those animals, how they are treated in those. And this is what we are also going to learn in this lecture as well, that there are very strict regulations if you have to use any animal. And for that, you have to be like, there are very strict regulations. For example, let me share one example with you. I was working in University of Alabama in Birmingham back in 2013. And there, we were doing research on cartilage. We were working on stem cells and regulation of stem cells in the cartilage development and role of microRNA in the regulatory in like how those stem cells can be differentiated into cartilage, and if there is some defect in microRNA, what could be the result of that? So, like we had an animal model there, and we were doing the research on the cartilage, and there was a whole group who were working on the hormones of that animal. There was another group that was working on the neurons of those animals. There was another group who was working on the skin of that animal. So we were using like a single mouse, and when that mouse was like sacrificed, then everybody was like rushing to the place that they can get their respective samples. So one could get like the skin, another can get the brain sample, another could get the sample from like the internal organs, another can get the blood samples, and we used to get the cartilage. So this is like how we can reduce the number of animals, and we can reduce the suffering of those animals, and they are properly regulated. So wherever you are working, similar was the case in Japan. So in Japan, there was like similar uh, situation. So we like whatever study we were doing, it was properly regulated and it was approved by the ethical review board of the institute, and only then we can start the study. Otherwise, you can't start. In Pakistan, still we don't have that much strict regulation, but now if you have to publish that work, and if that like for any publication, they ask that about the ethical approval uh, of that research. So for that, like for all the institution, there is institutional review. So for example, whatever is the research you are going to conduct, and there are a number of universities now in Pakistan. All the universities, they have the institutional review committee, which review your procedures and protocols. And if like they need to be like certain changes, they ask you for those changes, and then you can proceed for your study. So in Pakistan, even now, there are institutional review and there is institutional regulatory body which regulate all the protocols and procedures, and only then you can start those. And this is, and the third part is compliance and enforcement. So regular inspection and audits help ensure researchers adhere to regulations and maintain high standard of animal care. So even the animal laboratories, for the pharmaceutical research or for the biotechnology research, we use a lot of animals. So even those laboratories, they are fully like accredited that they follow the protocols and procedures to do research with animals. So all the diagnostic work you are using doing in your laboratories is actually done before on human. It's mainly done on the animals. For example, the new era which is coming for the diagnostic is the diagnostic through microRNA. I'm not sure if you are like working on those microRNA based diagnosis, for example, for the cancer diagnosis, for the diagnosis of diabetes, for the diagnosis of renal diseases and liver disease, for the cardiac diseases. So there were like old markers which were being used for like decades. But now the research is shifting that we can actually really see the stages of those diseases, and there is a shift of work towards the microRNA. So microRNA, you can identify those microRNA. So in order to do those diagnostic work in your laboratories, that work is being done on the animal first, and then that is translated into your diagnostic services. So microRNA is very much coming. Then exosomes are very much coming in diagnosis as well as for the treatment. So this is like the recent work. So all that work is being done on the animals first, and then that goes uh, for the diagnostic in the humans, and that is properly regulated. So like we can get, diagnosis is so important that if you set a protocols, if it is working in China, for example, and if it is not working in Pakistan, then how much harm we can have. Like we are getting the results there. So there may be some like problem, and all the institutions should have the similar protocols and similar finding, so that can be a universal protocol for the diagnosis. And that's why we go for the good laboratory practices, that all the things either going on in China or Japan or in Germany or in UK or US, they have the similar finding. If we also repeat the things back in Pakistan, so this is how we proceed for the animal studies to the human studies. In this slide, I will share with you that what could be the alternatives to the animal testing, and this is like very much focused now. Lab on the chip, for example, you have heard about. This is like very much coming even for your diagnostic work. You can run all the trials on the same chip, and you can do a lot of diagnostic based on that's the new like finding called the lab on the chip. So the whole like diagnostic work you can do either it's a DNA based or proteins based. You can do on a chip and you can test those things. So this is actually very much now into the fashion, and we can also use like other alternatives to the animals. Like we can use the computational modeling. So you know this is the era of artificial intelligence. This is the era of computational. This is the era of bioinformatics. So we can actually just like if you have to run the PCR, you know we can also run the PCR in silico. We can in silico we can get the results and we can see that if it is working on the humans or not. So like if you have some primers and you have to test those primers, instead of working on the animals, you can directly work on the humans. You can do the in silico work, and in silico you can get the like results whether it will be working on human or not. So we can do a lot of work now through the bioinformatics or through the computational modeling. Then another very much approach which is coming towards like the treatment and diagnosis is the microdosing. So microdosing, what as the name indicates, is like administering very small amount of the substance to the human volunteers and see that whether we don't directly test any drugs on the humans. We have to test on animals, but we can do the microdosing, like we can do some in silico work through bioinformatics, and then we can use very small quantity, which could be non-toxic to the human, and then we can see the behavior of the cells. And even now we can like have a single cell analysis. So we put some drug on the skin cells and we can see if there is some change to the skin cells or not. So now there is a lot of advancements, and this is also called like microdosing, that we can use a very small quantity and see whether it has some effect or not. Then we can also plan some epidemiological studies, so we can analyze population level data, and that can often yield valuable information without the need of animal experimentation. So we can uh, go for like large epidemiological studies, and from that we can also test that whether we can proceed for the animal testing or not. So this is how we can also save those animals. And the last thing is the tissue engineering. So this is very important approach. Actually, I can talk for hours on this because my postdoc work was on tissue engineering, and this is very much coming into the future. For example, for the cancer drugs, they have a very strong side effects on the humans. So what we can do in tissue engineering, we can develop some tissues into the organ. For example, if you have to develop the drug that can treat the brain cancer, so we can develop the organoids, uh, neuronal organoids into the laboratory, and we can test those drugs and see whether they has effect. So like it's not a 2D kind of thing; it's a 3D. So 2D is like we grow the cells, we put the drugs, and then we see the effect of that drug, like the tissue culture kind of research work we are doing. But in 3D, what we can do, we can actually grow those cells in a 3D model. For example, we can grow a miniature heart that is a beating heart in the laboratory, or we can grow piece of a liver in the laboratory, or we can piece of a renal organ into the laboratory, or a piece of a bladder in the laboratory, or a piece of a skin in the laboratory, and then we can test the drugs on those. And this is like very much humanized kind of thing. So in that case, we specifically know whether the drug is like acting on the cells or not. So just like I was giving the example of cancer, so in order to like treat the cancers, either it's like colon cancer, it's liver cancer, or it's a breast cancer, or I say neuronal cancer, we can grow those piece of the tissues in the laboratory, and we test the drugs and then see, so many there are so many drugs which are resistant, like even the cancer drugs, they are resistant in the human. So in this case we can get the cells and we can grow the tissue into the laboratory, and then we can see whether the drug is will be working on that human or not. So like how much time and effort and the suffering we can save in this case when we grow the tissues into the laboratory and the test the drugs, and like we can test so many drugs in a ray, and then we can see that this drug might be very good to cure the cancer and the other drug may not be working, that could be like have some kind of resistance. So this is how this is a very very good approach of tissue engineering which we can, and this is like this is the latest approach. I'm sure you must be familiar with these kind of research, and for that even we also need like all the regulatory requirements. So we should follow the regulations, and we should follow the GLP. If like some drug is being tested in Chokohana, that should be giving the similar result of the drug that is being tested in the city of Hope in California, US. So the results should be similar, so that can be replicated all over the world. So this is how we can like proceed for that. Then we have to focus on three R's for the animals. That is replacement, just like we have discussed in our previous slide about the replacement that we can use. The epidemiological studies. That's one thing we can go for. Bioinformatics. We can go for 3D tissue engineering, 3D like growth of the tissues. We can go for the lab on the chip kind of things. So in this case, we can replace the animals, and then the other thing is reduction. We can reduce the number of animals that are being studied. So reduction means, just like I have given you the example in the previous slides of my like one of the work that I was doing in University of Alabama in Birmingham. So this is how we can also reduce the number. So you can like collaborate with other researchers who are going to do the similar kind of study or who are using on a similar animal. So instead of like working on 10 animals, you can proceed with a single animal, and you can get all like all the researchers, they can get their respective sample, and this is how you can proceed further. And then the last R is refinement. So refinement is like we have to improve our procedures so we can go for minimize pain, distress, and suffering of the animal. So we can refine the procedure that, and we will definitely study more about the this refinement, that how we can go towards refine. I think some mic is on from the participants, so if they can mute it, so let me mute it myself. So you have learned about the three R's. That is replacement that we have just discussed. Reduction, I have given you the example, and I'm sure it will be very clear for you after that example that how we can reduce. And refinement, this is like how we can refine the procedures uh, so that uh, we can like improve the overall situation. So they should, and this is what we are also going to discuss about the refinement. You will have a better idea how we can do the refinement in our experiments. So housing and care standard for the lab animal, this is very, very important, especially like in all the developed countries, they take care so much. Like if you're going towards the animal house, they are like there is a proper temperature there, proper humidity is there. If you have to get into the animal house, you dress properly, you have a like head cover, you put on your mask, you have a lab coat, you put on the shoe cover. So you shouldn't take any of the like pathogens from the outside environment into the laboratory, into the animal house. And similar is the case like if you are moving out of the animal house, you remove all the ppes, so the pathogens and shouldn't be like move out with you if you are moving out of animal. Especially the first case is very important if you are working on some sensitive animals, like some kind we work on the animals which are immunocompromised. In that case, we actually follow the PPEs so strictly that we shouldn't take any of the pathogens from the outside inside the laboratory that can harm to the animals. So, what are the like in case of housings? What are the requirement? We should have like spacious enclosures. We should have a proper animal house where there should be proper space. It's like not like if you're working on 100 animals, for example, and you have a very small space that put a lot of pressure to the animals. So it should be spacious. Then we should also try for the environmental enrichment. So now, like there is also, just like we are providing a rich environment to our kids. Similar, we try to provide a rich environment to the animals. Like we have something which could be attractive for the animals, like some toys with them they can play. We have a proper bedding. We have like other stimuli that can promote their mental and physical well-being. So if they can like move around and kind of things. So this is like very much now followed all over the world. We should also try to focus on the specialized diet. You know, each animal has a different diet requirement. If you're working on mouse, if you're working on rat, cats, on rabbits, on monkeys or chimpanzees. So in all those cases, there is a proper diet, and we should also follow those proper diets. Even if you are working on nutrition, then you should like work in a way that you should provide them important nutritions, and then that's part of your protocol that is approved by the ethical review committee, and then you can proceed with the research. In some of the research is like very for example, some of my colleagues, they were working on like animals, and they have to burn their skin. So these are like procedures we should plan the research in a proper. So they were working on the burn treatment. Some of the researchers, they were working on the eyes. So like they have damaged the eye, and then they try to cure the eye. So like sometime even when they damage the eye, that cause infection to the eye, and then that can result in the death of the animal. So there is a lot of suffering. So in that case, there should be proper veterinary care. So if you are having animal house, there should be veterinarian there. So if they like there is some infection, he knows that what kind of antibiotics he has to give to the animals to stop the infections, or if you what that can be done to reduce the suffering of the animal. And this is being practiced in almost like I have worked in different countries, in Pakistan, also in US, in Europe, also in Japan. So in all those places, there was a proper veterinarian. Even I was working in Punjab University, and there was an animal house, and we have a like licensed veterinarian who was working with us for all the protocols. So like if there is any harm to the animals, he can do something. Like he has to do some bandages. If he has to, even like if he have to get samples. Sometimes we are so cruel to the animals to get those samples, but if we have a veterinarian with us, he knows how to handle those animals. So they can handle them very carefully. They can get the sample for us very carefully. So this is how we should involve those veterinary expert, and that's also part of like good laboratory practices. Like if you have an animal house, and if you have planned some study, you should involve a veterinarian with you. So there should be less and less harm to the animals during our research. So the next thing that we are going to discuss is about how we can minimize pain and distress in the animal experiments. So we have to work with animals sometimes. Just like I mentioned, for the last 20 years, I'm working with animals. We have to get the stem cells from them. We are working on some therapeutic proteins. We were working on tissue engineering. So in all those cases, we have to work with animals. But we have to minimize their pain and distress, and how we can do one of the thing is like anesthesia and analgesia. So anesthesia is very important. So if you have to get some samples, for example, we can use anesthesia. That could be local anesthesia, or that could be a general anesthesia that we have to give to the animal. So we can uh, reduce the pain, just like we reduce the pain in like procedures in the human. Similar, we should try that we should use the anesthesia in case of animals, so we can reduce their pains. We should also go for the early intervention. So, like sometime the animals they need veterinary care, just like I was discussing in the previous slide. So we should go for early intervention. For example, I was giving you the example. One of my students, she was working on the eye uh, diseases of the animals, and she was working on rabies. So in her first attempt, so when she was treating, there was an infection, and that result in the death of the animal. So like in all those these cases, we have to work very carefully, because if some veterinarian is involved there, we can reduce the suffering of the animals and also the distress. And then another terminology, euthanasia, that's also like we should humanly ending animal life. So after the experimentation, if the animal is suffering, we can end their lives. So the animal shouldn't suffer for a longer time. We can go for end of his life. So we can reduce the suffering of the animal, and we can reduce the distress of the animal. So that's very very important. So this is how we can minimize the pain and suffering. And so at the end, we are going to discuss about the role of institutional animal care and use committee. So just like we have discussed, we have now in all of our universities, just like we have the uh, uh, Rawalpindi Medical University or University of Health Sciences or all the medical universities. So they have the Institutional Animal Care and Use Committee, IACUCs. So they can go for like ethical review. So whatever procedure we are going to do, whatever study we are planning, either about the future protocols, as we have discussed, that in the future we are very much working on the, for example, microRNA, or we are going to work on the mRNA based vaccines, or we are going to work on exosomes. We have to like go for the ethical review of our protocols that we are going to work with those animals. And then they also, this committee, they also go for like monitoring. They do all the monitoring, that is not only in the papers that you said that you will go for this protocol. For example, you in the protocol you have mentioned that you will go for anesthesia, but when the committee go and see you don't have any anesthesia, and you are not following those anesthesia things in your practical work, that's against the regulation. So they also go for oversight and monitoring of our your protocols. And lastly is the continuous improvement. This is like part of good laboratory practices that we are discussing for the last so many like lectures, that in all the protocols, in all the procedures, we should go for the continuous improvement. Just like I mentioned that we can replace the animals. So if there is a way that we can replace the animal, this is part of your continuous improvement. So the committee, they work with the researchers to identify ways to refine procedures and reduce animal use whenever possible. This is like what we have also discussed, that this is how we can reduce the animal suffering uh, with the continuous improvement. Like if there is a way that we can switch from the work on the animals to in silico kind of studies, how we can do that and how we can improve the overall protocols. So conclusion is upholding the highest standard of animal research ethic. This is very, very important. So by adhering the rigorous ethical guidelines, as this is just like I mentioned, this is part of our good laboratory practice, that we should follow the rigorous ethical guidelines. We have to maintain strict oversight for whatever protocol we are doing, and continuously exploring alternatives. The scientific community can ensure that the animal research is conducted with utmost care and respect for well-being of all living creatures. So the work we are doing with the animals, it should be like properly work, and all the things almost we have discussed. So starting with the animals, that we if we have animal house, there are properly guidelines now for those animal house, about the temperature, about the like humidity inside the animal, about the diet, about the like quality of the water, everything, and the presence of a veterinarian there. Then whatever like protocol you have devised, there is a committee that actually review those protocols and also monitor your research work, that it is done with utmost care and without like uh, wasting those animals. So sometimes our researchers, like the research, if I share with you that in some cases we are using a lot of animals, and we are like instead of using all those animals, we can reduce their numbers. But like sometime if you're working on a neurodegenerative disease and we have to sacrifice like hundreds of those animals on which we are doing the research work, we can minimize their number. So, and they should be like properly. If you're working on, if you have to remove the brain of the mouse, for example, so what you can do, you can uh, use proper anesthesia. So, for example, let me share with you one example. So, if you have to take the blood from the animals, so there is like two ways. If you have done, for example, anesthesia, that can actually uh, that can sometimes kill the animal. So, even the higher dose. So, you have to choose the dose properly. So there is one way, like protocols, you can go and directly inject the needle into the heart of the mouse and you can get the blood. And there are other ways to get the blood. So we should follow those protocols which actually give minimum harm to the animals. If you have to get the sample, like if you have to get the sample from the mouse, and if you take extra blood that can harm the animal, you can reduce the number of and volume of the blood that you're. For example, if you have to get, for example, uh, um, 1 ml of the blood, you can reduce it to like a half ml, and you can try to do the research work on that. This is how we can actually minimize, and then we should like properly follow the guidelines. These are also very, very important for your publication. So with this, I think our time is also going to over. This is, and we have to plan one or two more lectures. Probably this week we will try, and I request you that if you can spare some time, we can plan some lecture during the week, or if you like want to